After completion of chapter number 8 and 10, means the cell and cell division, now we are going for the learning of chapter number 9, and that is biomolecules. Biomolecules present within the protoplasm of cell. These biomolecules, which is categorized into two types, inorganic molecules as well as the organic molecules. Generally, carbon containing organic molecule present in a protoplasm is considered biomolecules. But in protoplasm of cell, not only just organic molecule present, along with inorganic components also there. These inorganic components, which is two types, one water and second minerals. Minerals from soil, first of all, absorbed by plant. With respect to how much requirement of these minerals in plant body, these minerals categorized into two types, macronutrients and the micronutrients. We have to further learn what is micro and what is macronutrients, their examples and their important role in plant as well as animals, and the deficiency symptoms. Organic chemicals, which are carbohydrate, lipid, protein, nucleic acid. There is two types of nucleic acid we know. One, DNA, that is deoxyribonucleic acid, and second, RNA, ribonucleic acid. Enzymes and hormone. Majority of hormone which is made up of protein, but some also made up of lipid. Enzyme most often made up of protein. Some nucleic acid also act as an enzyme, but there is few examples. So enzymes as well as hormone, that is also organic chemicals. So our body, organism having a carbohydrate, lipid, protein, nucleic acid, enzymes and hormone, these are organic chemicals. Out of all, we are first going for the learning of carbohydrate. There are few examples and types we already have. First, our organic chemical, that is carbohydrate. Carbohydrate, which is made up of carbon, hydrogen and oxygen. Here ratio of hydrogen to oxygen, that is 2:1. Means 2 hydrogen at that time, 1 atom of oxygen in composition of carbohydrate. General formula for the carbohydrate. General formula for the carbohydrate, that is CN H2 in bracket M. Here in carbohydrate, value of N is equal to M in some carbohydrate, whereas other carbohydrate N not is equal to M. For the construction of any carbohydrate, their general formula CnH2O whole bracket M. Here value of N equal to M or not. Carbohydrate categorized into three types: monosaccharide, disaccharide and polysaccharide. Monosaccharides, disaccharides and polysaccharides. We take their example. For example, glucose is the example of monosaccharide. 2 glucose joint produce and form maltose. So maltose is the example of disaccharide. Many glucose molecule link to each other and producing polymer of monosaccharide, that is starch. So starch is the example of polysaccharide. We can reversely also think starch hydrolyze producing maltose, maltose hydrolyze producing glucose. Now, out of this all different types of carbohydrate, we are first going for the learning of monosaccharide. So first write their formula. In general formula of carbohydrate, A is equal to M if we take. So that is monosaccharide formula. General formula for monosaccharide is CN H2O N, because here N is equal to M, so either write NN or MM. So this is modify and write CH2O whole bracket N. Here value of N, that is 3 minimum, N is equal to 3 up to generally maximum 7. So for monosaccharide, N is equal to M. So for monosaccharide formula, there is CH2O whole bracket N or M. Value of N minimum 3 to 7. Monosaccharide example glucose. Monosaccharide is simplest sugar. That is simplest carbohydrate. Why we can say sugar? Because sweet in taste. Sweet in taste. That's why there is can say sugar. Monosaccharide can say sugar. Monosaccharide glucose dissolve in water. So we can say monosaccharide which is soluble in water. As well as any monosaccharide easily cross the plasma membrane because dissolve in water. So characteristic or property of any monosaccharide which is same as sugar, sweet in taste, soluble in water, easily cross the plasma membrane. Now see, starch hydrolyzed so produce maltose, maltose hydrolyzed to produce glucose, means polysaccharide hydrolyzed into starch, polysaccharide hydrolyzed into disaccharide, disaccharide hydrolyzed into monosaccharide. But monosaccharide not further hydrolyzed, means glucose not further hydrolyzed. Yeah, glucose going inside respiration, that is okay, but water molecule not directly enter in glucose and glucose not split into any component. So important characteristic of monosaccharide, not hydrolyzed. Now we are going for the discussion of now different type of monosaccharide on a base of what is the value of N. 3, 4, 5, 6, 7. If you take 3, so here 3 carbon, so that is triose type of monosaccharide. 4, so C4. Tetrose 5, pentose 6, hexose 7, heptose. So according to number of carbon, monosaccharide various type. Triose, tetrose, pentose, hexose, heptose. As well as monosaccharide having a 2 functional group, one aldehyde and second ketone group. So according to aldehyde and ketone group, monosaccharide divided into 2, aldose sugar and ketose sugar. Now we classify the different monosaccharide and their example. Monosaccharide general formula that we know, clear cut, CH2O whole bracket N. N. Monosaccharide which is made up of 2 categories of functional group, one aldehyde group. So this monosaccharide is can say aldose sugar, and some other monosaccharide which is made up of ketone group, so that is can say ketose. Now first we take N is equal to 3. N is equal to 3, so we can get empirical formula of that monosaccharide, that is C3H6O3. 3 carbon containing monosaccharide is can say triose, and their empirical formula C3H6O3. Empirical formula means that indicate the number of carbon, hydrogen, oxygen, whatever present inside chemical. Now we have to prepare here triose 2 type, aldose type as well as the ketose type. We having 3 carbon, 6 hydrogen and 3 oxygen that we arrange inside their molecular formula. So write 3 carbon in sequence, and carbon having a valency 4. Here we preparing the aldotriose and here ketotriose with made up of 3 carbon. First carbon, second, third, first, second and third. Now we have to remember here aldehyde group always present first carbon of any monosaccharide and ketone group always present at the second carbon of any category of monosaccharide, triose, tetrose, pentose, hexose and heptose. Okay, now we have to arrange here 3 oxygen and 6 hydrogen. 3 carbon already arranged. Oxygen, first, second and third. Here, now arrange hydrogen. CHO group present at the first carbon. So this is CHO, CHO. Now rest of the 5 hydrogen we arrange. First already arranged, second, third, fourth, fifth and sixth. So this is one of the example of aldotriose. Now ketotriose. Ketone group always present at the second carbon. Okay, 1 oxygen only arrange. Now left 2. 6 hydrogen now arrange. Here we cannot arrange any hydrogen because oxygen valency already failed. So now 2 chemical we get. The name of this chemical is the glyceraldehyde and name of this chemical dihydroxyacetone. Glyceraldehyde is the example of aldotriose and dihydroxyacetone. Dihydroxyacetone is the example of aldotriose. So we complete here triose category of monosaccharide. Both molecular formula same but structural arrangement different. So any chemical their molecular formula same, C6 C3 H6O3 C3 H6O3 both, but structural formula different, so they are can say isomer to each other. Glyceraldehyde and dihydroxyacetone, they are isomer to each other. Now question is that why we learn this triose? Because this category of triose help in a chemical reaction for photosynthesis as well as the respiration. In photosynthesis which phase? So dark phase. But as it is chemical not used. Their third carbon phosphorus attached. So once again, both the chemical we write and phosphorus attached at the third carbon in place of hydrogen. Same chemical compactly you can write by this way, CHO directly. Then CHOH, CHOH. Instead of this you can write CHOH. And then CH2OH. So this is glyceraldehyde. Now dihydroxyacetone. CH2OH, C double bond O, CH2OH. CH2OH. C double bond O, CH2OH. This is dihydroxyacetone. The chemical take participate in dark phase and photosynthesis. There were third carbon, first carbon, second, third, first, second, third. Carbon phosphorus attached in place of hydrogen. So name of this chemical is phosphoglyceraldehyde. Phosphoglyceraldehyde, PGAL. And name of this chemical is dihydroxyacetone phosphate, DHAP. Da and pagal isomer to each other. This is phosphoglyceraldehyde. phosphoglyceraldehyde. And dihydroxyacetone phosphate. Dihydroxyacetone phosphate. Both the chemical where take participate. So both the chemical take participate in respiration as well as the dark phase of photosynthesis. Now we take N is equal to 4. So what is the empirical formula of monosaccharide? C4H8O4, and this is tetrose. Tetrose we have to prepare two types, aldotetrose and ketotetrose, as we prepare aldotriose and ketotriose. Which are the example of aldotriose? Glyceraldehyde and PGL. Ketotriose, dihydroxyacetone, and the okay, now we have to arrange one row of carbon which is having 4 carbon for aldose, every having a valency 4. Once again 4 carbon in a one row. For the ketose type of tetrose, first carbon, second, third, fourth. First, second, third, and fourth. Now always aldose sugar, aldehyde group present at the first carbon, and ketone group present always at the second carbon. One oxygen arrange and one hydrogen arrange. What is left? 3 oxygen and 7 hydrogen. Oxygen, oxygen, oxygen. 4 oxygen arrange. Now 7 hydrogen. Second, third, fourth, fifth, sixth, 7 and 8 hydrogen arrange. Which chemical completed? Aldotetrose completed. Now ketotetrose. 1 oxygen arrange. Now second, third and fourth, 4 oxygen complete. Now hydrogen, first, second, third, here cannot arrange, fourth, fifth, sixth, seventh, eighth. Ketotetrose completed. What is the name of this chemical? So that is erythrose. What is the name of this chemical? Erythrulose. So erythrose is the example of aldotetrose and erythrulose is the example of ketotetrose. These chemicals take participate in dark phase. Most of the monosaccharide who having a role inside the dark phase procedure means Calvin cycle. Now we take N is equal to 5. So we can get which type of monosaccharide? Pentose. Pentose sugar, their empirical formula C5H10O5. C5H10O5. This is empirical formula of any pentose. Now, many example of aldopentose as well as the ketopentose we have to write. So, mention one row of carbon having a valency 4 and carbon how many? 5. First carbon, second, third, fourth and fifth. all having a valency 4. First carbon, second, third, fourth, fifth. Carbon number first, second, third, fourth, fifth. First, second, third, fourth and fifth. Ketose group always present at second carbon. And aldehyde group present always at the first carbon. Directly write what? CHO. Now here left to arrange 4 oxygen. Here also left to arrange 4 oxygen. Now arrange hydrogen. Second hydrogen, third, fourth, fifth, sixth, seventh, 8, 9 and 10. This is the example of aldopentose. Hydrogen first, second, third, fourth, fifth, sixth, 7, 8, 9 and 10. This is ribose. This is which biochemical structure? This is ribose. And this is ribulose. Ribose present where? So ribose occur in a composition of RNA. Means ribonucleic acid as well as the energy currency of cell, that is ATP. RNA and ATP, which is made up of ribose type of pentose sugar. Ribulose where take part is separate. So ribulose as it is denoted, but there were first and the fifth carbon phosphorus attached. Okay, so we write the chemical. CH2OH, CH2OH. Directly write. Then C, double bond O, COH, COH, and C, H2OH. Hydrogen and hydrogen. So this is also same structure and this is ribulose. In the structure ribulose, their first carbon and fifth carbon in place of hydrogen, phosphorus 20. So name of this chemical is ribulose 1,5-biphosphate. This is ribulose 1,5-bi or diphosphate. So now this ribulose 1,5-bi or diphosphate in short form is can say RUBP या RUDP. This ribulose biphosphate where play important role. So this is play important role in a dark phase of photosynthesis. So ribulose as well as RUDP या BP are related to or example of ketopentose and ribose is the example of aldopentose. Ribose sugar, their second carbon, oxygen missing. So this is can say deoxyribose. Okay, now we write new structure by modification of oxygen missing from there. So CHO, H and H, oxygen missing at first, second carbon. Then rest of the chemical structure remain as it is. CH2OH. First carbon, second, third, fourth, fifth. In ribose structure, second carbon, oxygen missing, so this chemical is can say deoxyribose. deoxyribose. And deoxyribose present in a structure of deoxyribonucleic acid, RNA, DNA. RNA there were ribose present, DNA there were deoxyribose present. So pentose sugar, especially aldose sugar, aldopentose type of sugar present in a structure of nucleic acid as well as the ATP. Now we are going for the discussion of isomer. What is isomer? So same molecular formula, the chemicals who having the same molecular formula but structural arrangement different, so they are can say isomers. This isomer also divided into two, anomers and epimers. What is this? We write first their example. First we write the ribose isomer. There is a two isomer of ribose. Now see these three chemical which is exactly same. All at presently there is ribose. But if at the second carbon position of H and OH change, OH going from right side to left side and H left side to right side. So this is the new chemical produced and that is the isomer of ribose and that is can say arabinose. How to produce arabinose? By changes of H and OH position at second carbon. But same changes at the third carbon, H going right side and OH going left side. So that is can say xylose. So xylose, arabinose and ribose, they are isomer to each other because their molecular formula C5H10O5 but structural arrangement different. And see here this isomer formed by changing of carbon, both the side hydrogen as well as the oxygen, except the first carbon. If isomer formation, except the first carbon, any other second, third, fourth, any other carbon, H and OH position change, so there is epimer. If first carbon H and OH position change and producing isomer, so that is can say anomer. Here we not write any example related to anomer, but in our next study we will also mention the example of what is anomer. So now isomer divided into two, anomer as well as the epimer. What is epimer? So isomer formed due to changes of H and OH position at the second carbon, third carbon, fourth carbon, whatever, except the first carbon. So originally ribose, arabinose and xylose are the epimer to each other. See here molecular formula of deoxyribose change because one oxygen missing. So we can mention C5H10O4. As we write ribose epimer, also we write ribulose epimer. This is the structure of ribulose. This is also the structure of ribulose. Changing at the third carbon, position of H and OH. So we receive here epimer of ribulose and that is xylulose. This xylulose also participate in the phase of photosynthesis. So we complete here three types of at presently monosaccharide: triose, tetrose and pentose. And all once again two type: aldose sugar and ketose sugar. Next lecture we will discuss about two hexose and heptose, means monosaccharide medium of 6 carbon and 7 carbon number. At presently, which are the example of aldose sugar? So glyceraldehyde, PGL, erythrose, deoxyribose, ribose, arabinose, xylose, all are the example of aldose sugar. At presently, which are the example of ketose sugar? Xylulose, RUBP, DP, ribulose, erythrulose, DA, as well as the dihydroxyacetone. See this information, whatever we learn in a monosaccharide, which is not given in textbook. So you have to write directly from board. Not read from textbook because not given any information from there. Our textbook only mentioned carbohydrate. There were few little bit information about to polysaccharide just. So when polysaccharide came, that topic at that time open the textbook and read from there.